Hello, my name is Dr. Katie Snape. I am a consultant cancer geneticist from St George's Hospital in London, and I'm your lead educator on this course, Interpreting Genomic Variation, Inherited Cancer Susceptibility. Thank you very much for joining us on this programme. This course has been specifically designed for healthcare professionals and clinical scientists who are interpreting or discussing the clinical relevance of genetic variation in cancer susceptibility genes. So we do assume a high level of prior knowledge and use scientific language throughout. We would strongly recommend that all learners on this course have previously completed the Fundamentals of Variant Interpretation course, which is also available on the Future Learn platform, or who are familiar with the principles of variant interpretation through their clinical practice. The aims of this course are to critically appraise the strengths and the weaknesses of the American College of Medical Genetics, the ACMG guidelines, when we're thinking about variant interpretation in complex diseases such as cancer compared to rare pediatric diseases. And this is really possible through the embedding of large-scale collaborative infrastructures to standardize evidence-based variant interpretations in cancer susceptibility genes. And specifically in this course, we're going to be looking very closely at the guidelines that have been produced by the CanVig UK group of scientists and clinicians who've set out to standardise variant interpretation in cancer susceptibility genes in the UK. And we'll be particularly focusing on hereditary breast and ovarian cancer and inherited colorectal cancer such as Lynch syndrome. By the end of the course, we hope that you'll be able to critically appraise the complexities of applying cancer susceptibility gene variant interpretation in clinical practice, and also that you have some idea of future complexities and changes in this rapidly moving field. So this will be the beginning of our education in this area, but I am absolutely certain it will not be the end. Look forward to seeing you throughout the course. Please do interact with your fellow learners. If you feel comfortable on this step, please give us a little bit of information about your background, what your previous clinical experience is, why you are coming to do this course, and what you're hoping to achieve from it. And we look forward to having future discussions throughout the course. So hello, welcome to week 1 of the course. In this week, we're first of all going to really think about why variant interpretation in cancer susceptibility genes is different to variant interpretation in rare disease, what some of the complexities are when we're considering common and complex disorders, but potentially what some of the advantages are as well when we are dealing with genetic variation in a phenotype which is more common within the general population. So we're going to think about large-scale infrastructures that can help us interpret variation in cancer susceptibility genes, and we're going to start applying some of the principles of the CanVig UK guidelines, specifically thinking about lines of evidence relating to population data and to in silico tool data, and we'll be doing that through the use of lots of case-based studies. So look forward to discussing and interacting with you throughout the week as we do that.
What are cancer susceptibility genes? We have already considered how cancer is a complex disease with a range of possible causes, some inherited, others not. We know that a small proportion of all cancers are caused by a rare, high or moderate risk genetic cause. This is a pathogenic variant in a cancer susceptibility gene. Pathogenic variants in cancer susceptibility genes are known to increase the risk of a person developing cancer over their lifetime. The exact risk and types of cancer vary depending on the specific gene. When we talk about cancer susceptibility genes, we are specifically referring to genes in which a single pathogenic variant is sufficient to increase the lifetime risk of developing cancer by at least 2-fold. Although the risk is usually much greater than this. We are not including low-risk genetic variants which increase the risk of cancer by small amounts, less than 2-fold. which have been identified through genome-wide association studies and which confer cancer risk in a multifactorial way. Whilst cancers which occur due to an inherited pathogenic variant in a cancer susceptibility gene account for a relatively small proportion of all cancers, it's important to identify individuals who have this significant genetic risk of cancer. Offering these individuals screening, prevention and early detection programs tailored to their specific cancer susceptibility gene and their lifetime risk of cancer improves clinical outcomes and is cost effective. In most cases, a person only needs to inherit a pathogenic variant in one copy of a cancer susceptibility gene to have an increased risk. This means the inheritance pattern is autosomal dominant. However, there are some rare autosomal recessive conditions with increased cancer risk. We will consider these briefly later in the course. Historically, cancer predisposition genes were identified through studying families with a strong history of cancer. However, more recently, more wide-scale germline and somatic cancer gene testing has led us to the identification of an increasing number of pathogenic variants in cancer susceptibility genes, both in cancer patients and in population-based genomic studies. Many cancer susceptibility genes are those involved in key steps to carcinogenesis, be this DNA repair mechanisms or cell cycle regulation. Throughout this course, to illustrate the key concepts in variant interpretation in cancer susceptibility genes, we will focus primarily on the two common cancer susceptibility syndromes. Firstly, hereditary breast and ovarian cancer caused by variants in the BRCA1 and BRCA2 genes. Secondly, we will consider Lynch syndrome, an inherited cause of colorectal, endometrial and other cancers. Lynch syndrome occurs due to variants in the MLH1, MSH2, MSH6 and PMS2 genes. So thank you both for coming to your genetics appointment today. I've got the referral from your GP and I understand, Liz, you have some concerns about your family history of cancer. But to begin with, it would be helpful to hear in your words why it is you come to genetics today.
Yeah, we've come today because we want to start a family, and I'm struggling to think about that before I find out a bit more about the genes sort of that might run in my family, because my mum was diagnosed at 39 with breast cancer and died 6 years later. Her auntie had ovarian cancer and died around 50, and her grandma got breast cancer when she was in her early 40s as well. So people say there's a curse on the family, and I just want to know what the possibility is that I'm going to pass that down to my children, because I don't want that to happen to them, really.
And if we do find that out, mastectomy's not off the table, is it? If that's something that needs to happen, and we do want to start a family, so it would be good to know, because we wouldn't want anyone to have to go through what you've gone through, really.
No, and I'm 37 now, and so Mum was diagnosed at 39. So it's not many years away. And when she spoke to the doctor, she did mention me, and she said that she left a blood sample for research, but we didn't get any results from that, so we don't know if anything's happened to that. So we just want to know what the possibility is there.
And you've not had breast screening or checks?
No, nothing.
So Liz was 12 when her mum got breast cancer, and 19 when she died. So we certainly wouldn't want to. Be a lot to bring another child in with the possibility of that happening to them, so we wouldn't want to do that.
Okay, so thank you both for sharing all of that with me. It sounds like it's understandably been difficult for you, Liz, and that your risks are something that plays on both of your minds, in particular when it comes to starting a family. So to begin with in today's appointment, I just wanted to thank you for filling out all of the information about your family history, and with all the details you gave us, we were able to contact that research study that your mum had taken part in. And you were completely right, there weren't any genetic results from that, but there was a stored sample of her DNA that we would be able to use to do some genetic testing. So we can talk about that test in a bit more detail, but to begin with it would be helpful just to go through the family history, also find out a little bit more about both of you, and then we can go on to talk about the genes that cause breast and ovarian cancer risk, and we can talk about doing that genetic test on your mum's stool sample.
Thank you.
Thanks.
Cancer and animals. Up to 1 in 2 humans will develop cancer in their lifetime. That's half of us who will one day develop genomic changes that transform our cells into deadly invaders. But this isn't true for all animal species. Whilst myths abound that sharks never get cancer, this isn't actually correct. Sharks do appear to be more resistant to cancer than humans, but scientists first noted cancerous tumours in sharks over 150 years ago. Over 40 different benign and cancerous tumours have been identified in at least 18 different species of sharks. Persistence of the myth appears to go hand in hand with sales of shark cartilage in alternative medicine, and probably plays a role in endangering certain shark species. However, there are animals who don't get cancer. The naked mole rat may look like it's just been pulled out of a burrow backwards and hit with the ugly stick, but scientists have not observed any naked mole rats developing cancer in long-term studies of naked mole rat colonies. Their secret is thought to be the secretion of a special type of hyaluronic acid within their cells. At the other end of the size scale, the bowhead whale weighs in at 100,000 kilograms and has around 3.7 quadrillion cells, which is about 1,000 times more cells than humans. Yet, for all their many cells, they are resistant to developing cancer. The bowhead whale genome was sequenced in 2016 and is still being interrogated in the hope of finding the secret of the animal's success. Finally, although around 3% of elephants get cancer, this is significantly below the 50% cancer rate in humans. Scientists have discovered that elephants have at least 20 copies of the TP53 gene, known as the guardian of the genome, for its role in cancer prevention. This is a lot compared to the 1 copy found in humans. Whilst we're not likely to get down to elephant rates anytime soon, the hope is that by understanding the genomes of animals who are more resistant to developing cancer, we will find new insights which might help treat or prevent human cancers.
My name's Stephen Hardy. I'm Head of Molecular Diagnostics for the National Disease Registration Service in Public Health England. So I lead on the end-to-end delivery of this programme, which includes working with labs and data providers to collect data at source, through curation, quality assurance and processing within the department, right through to analyses and various outputs at the other end. So my involvement with the CanGene CanVar programme is as co-lead of Work Package 1A. So Work Package 1A is focused on the collection and the amalgamation of germline genetic testing data from the laboratories within the Genomic Medicine Service. Within the National Disease Registration Service, we collected on all individuals who receive germline genetic testing for cancer susceptibility within the NHS. So some well-known examples of such testing would be BRCA1 and 2, or the BRCA genes, which can lead to hereditary breast and ovarian cancer, or mismatch repair testing, which is a group of genes which can lead to Lynch syndrome. In terms of the information we collect on the test, we collect a lot of really important information. We collect information around the type of test. So was it a diagnostic test? So this is testing that's performed in the proband or the individual who presented to clinical genetics, quite often cancer patients themselves, or was it a predictive or pre-symptomatic test? So this is testing performed within a family member where a genetic cause of cancer has been identified in another family member. and that testing is performed to help clarify their risk of developing cancer. We collect information around the outcome of the test. So did we or did we not detect a sequence change or a sequence variant that we believe might be relevant or causative of the patient's condition? And if we did detect a sequence change, we collect very specific, really precise details on the nature of that sequence change that was detected. And the reason this data collection is so important is that for the very first time, we're actually able to collect and pool together all of the data from all of the labs that perform this testing on a truly national scale. And that's really, really beneficial in helping us to better understand genetic test results and what they mean for patients and their families. So we collect the data from all of the labs that perform this testing within the NHS. So it is a truly national data set, which is a really phenomenal achievement. And it has been a huge national initiative. You know, the labs have really worked hard to provide this data to us. And of course, the patients themselves as well are really key and central to this as well. So the way we collect the data, we use a pseudonymization based approach. So by pseudonymization, what I mean is we take a data record and we make it less identifiable. So any information that can be used to identify a patient, such as name, date of birth, NHS number, postcode, anything that can be linked to a specific patient, we actually remove and we replace it with a pseudonym, which is essentially an artificial identifier specific to that patient. And that's really, really important because that, you know, helps keep the patient data safe and secure, which is absolutely our number one priority. What isn't pseudonymized is the variant information. So the variant information is still visible. So that means we can look at those aggregate variant counts and release those back to the clinical community via the CAMVAR UK database. I mean, the major benefits of this, I think we're already actually starting to realize the benefits of this. You know, Kanji and CAMVAR, it's still in fairly early stages. You know, we're 2 years in now, and I think we're already starting to see major benefits from this data collection. You know, there's a number of examples where from the aggregate variant counts that we've produced from the national data sets that we're actually able to do case control comparisons. And we've actually been able to take that as a piece of evidence in its own right to change the classification of variants. So from a variant of uncertain significance, which is, you know, clinically unactionable, you know, probably the biggest challenge we face in clinical genetics is variant interpretation and how we deal with these, you know, variants of uncertain clinical significance. You know, there's numerous examples where the national collection of this data has allowed us to reclassify a variant both ways, either into a benign category or into a pathogenic, likely pathogenic category, both of which is useful from a clinical perspective. And linking the genotype to all of the other really rich clinical data we have within disease registration as well. So in terms of the National Cancer Registration Service, which is part of disease registration, we have the most complex, detailed data collection probably in the world. And I think the ability to bring in that genotype data and link it to pathology, to treatment, to outcomes, you know, it really allows us to look at how genotype is influencing phenotype, treatment and outcomes on a pretty unprecedented scale. I'm really excited to see how the data is going to change and develop over time. So our initial focus to date has been very much on those common genes that are commonly tested within the lab. So BRCA1 and 2, hereditary colorectal cancer syndrome, such as Lynch syndrome. I think in terms of how it will change, it will get bigger. So we're working really hard with the labs to collect retrospective data as well as prospective data. So to put it into context and give you an idea of the kind of size and the scale of the data we have. So for BRCA1 and 2, we have around 150,000 tests collected from the labs going back over a time period of 20 years. So it's a really large data set, growing all the time. And the power is really in the size and, you know, those big numbers. So I think the data set will get bigger. I think it will get better. You know, we're always looking to improve the quality of what we do and the data we provide to the clinical community. And I think it will expand. We're already beginning to expand our data collection to the other cancer susceptibility genes as well. So, for example, TP53, VHL and others. The ultimate aim is to collect complete representative data on all cancer susceptibility gene testing within the NHS. And we're really well on our way to doing that.
So just to recap what we've talked about already, we've talked about what genes are and how sometimes there can be changes in genes that can cause them to not work properly, and sometimes this causes an increased risk of cancer. And we focused on 3 genes in particular. The first 2 are the BRCA1 and BRCA2 genes that we know are linked to an increased risk of both breast and ovarian cancer. And we've also talked about the PALB2 gene, which we know mainly causes an increased risk of breast cancer. And so both of those categories would really fit based on your family history of people being affected with breast and ovarian cancer at a young age. So we talked about testing your mum's stored sample for those genes to see if we can find an explanation for the family history of cancer and for her diagnosis. And so next, I'd like to talk about the different outcomes that might come from this test. So there are 3 different possible results. So the first possibility is that we don't find anything at all that explains why there is this family history of cancer. And if this is the case, it does mean that it's likely that there isn't any of these high-risk causes of cancer in your family. However, it does mean that we haven't found an explanation for why there is that family history of cancer, and it means that there's nothing we'd be able to test you for, Liz. If this is the case, we would still recommend some screening for you, and that would be based on the family history as it is. So that's the first option. The second outcome is that we might find a change in one of these genes that we are confident explains why your mum developed cancer. And if we find one of these changes, we're also confident that it does increase lifetime risk of both breast and ovarian cancer. If this was the situation, we could talk Liz in another appointment in more detail about testing you for this change, and also if you did carry the change, what options would be available to you in terms of reducing your risk, but also family planning options.
Okay.
The first 2, yeah, they make sense. The third option is the one that's a little bit more complicated, and that's that sometimes we find a change in one of these genes that we don't fully understand, and we've all got changes in our genetic material, and they're just the things that make us unique. So if you were to look at mine and your DNA today, we find about 5 million different changes in there. And those changes are not things that we think cause genes to stop working properly. They're not things that cause an increased risk of cancer. And actually they're just part of normal variation. They're what make me me and you. And so we don't need to worry about those changes. And sometimes when we do find a genetic change, we can be really confident that it is just part of this normal variation. Other times, we've got lots of information and we're really certain that it does definitely cause an increased risk of cancer. But every now and again, we get this third option, this thing in the middle where we're not quite sure if it does cause an increased risk or not.
What would you do to work that out? Because that would be a really difficult situation for us.
Yeah, understandably. So when we find these changes, we call them variants of unknown significance. And when we find that, we work with our lab to see if there's any more information we can find to try and help us be a bit more certain either way. And we can also have a further appointment when we get that result and kind of talk about those next steps.
Okay, thank you.
The first areas of the CanVig UK and ACMG guidance that we look at in detail are how we can use population databases and case control data to help classify variants in cancer susceptibility genes. Using population data in variant interpretation can provide evidence across the spectrum of variant pathogenicity. Strong evidence that a variant is benign is provided if frequency within a population is higher than would be consistent with the frequency of a relatively rare disease-causing variant in a cancer susceptibility gene. However, using population frequency as evidence of pathogenicity in cancer susceptibility genes is more complex than it is for rare disease. This complexity arises from a number of different areas. Firstly, population databases are more likely to contain individuals who have had cancer than those who have had a rare disease. 1 in 2 people in the general population are diagnosed with cancer in their lifetime, and many of the studies used in population databases only exclude individuals with a severe or childhood onset phenotype rather than all medical diagnoses. There are multiple different control datasets, and it's important to understand which ones contribute to each population database and to align this to the case data where at all possible, i.e. identify if you can remove any confounding individuals that will bias the outcome. For example, if using the European control dataset and NOMAD for cancer susceptibility genes, it's possible to remove the Cancer Genome Atlas set. This cancer-free population data can be accessed through the CanVar UK website. For the purposes of this MOOC, however, we'll be using the UK Biobank population data to align with the UK National Disease Registration Service. In addition, the variable penetrance and later onset of disease associated with cancer susceptibility genes means that pathogenic variants in these genes may be present in population databases, even in unaffected individuals, as a person may carry a pathogenic variant but not have developed cancer at the time they were included in the database. Another factor that complicates the use of population databases is the relatively high frequency of some cancer predisposition variants in the population when compared to the vast majority of variants causing rare disease. The population frequency of pathogenic variants in all cancer susceptibility genes is estimated as around 1 in 200 to 1 in 500 in the general population, and can be enriched to even higher frequencies in specific populations due to the presence of founder mutations. For example, we know that there are 3 BRCA founder mutations, which can be seen in up to 1 in 60 individuals in the Ashkenazi Jewish population. All of these factors together mean that pathogenic variant and cancer susceptibility genes may be present in population databases. This makes it less likely that we're able to apply PM2, absence from population databases, for cancer susceptibility genes than for rare pediatric disease genes. However, if appropriate data is available, PS4, prevalence in affected individuals is statistically increased over controls, can provide evidence towards pathogenicity, even for variants seen as a reasonable frequency in the general population. We will look at PS4 in more detail in an upcoming step. As a result of this complexity, the CanVig UK guidance has suggested adaptations to the use of population databases for the interpretation of variants in the more common cancer susceptibility genes. These include assigning different strengths to the evidence for PM2. PM2 can be used as moderate when there are no observations of the variant in a control series of more than 50,000 individuals, as this suggests the variant is very rare in the population and so a higher chance of being pathogenic. They have also assigned supporting evidence where a variant is observed in 1 individual in a series of over 50,000, allowing for the increased prevalence of cancer predisposition gene variants in the population. However, they do not recommend applying PM2 at all if the variant frequency is greater than 0.002% in any population of any ethnicity. It's important to remember that the gene-specific guidance is also available for the use of PM2 in some cases. This gene-specific guidance can also be found through the CanVig UK website and includes specifications such as the use of female-only data when looking at population datasets for BRCA variants. Continuing our look at the CanVig UK guidance for using population databases in cancer susceptibility gene variant interpretation, we will now look in more detail at the ACMG category PS4. Prevalence in affected individuals is statistically increased over controls. To undertake statistical analysis of case and control data, we need large-scale NHS-wide data on the number of times a variant was identified out of the total number of tests performed. We also need this data for both cancer cases and from control populations. This data also needs to be accurate and ethnicity matched. This may sound like data should be easily available, but actually, until recently it was impossible to collect this. In the UK, diagnostic genetic testing was undertaken by a small number of independent NHS genetic laboratories, and the results were kept within separate lab databases. Each lab kept their own record of how many tests they had done and the variants they had identified. But there was not a systematic way of collating and sharing this data that also respected individual patient confidentiality. A new infrastructure was therefore required to get the large data sets needed for this type of analysis. A collaborative approach between the National Disease Registration Service, formerly known as Public Health England, the NHS Genomic Laboratories, and the CanGene-CanVar research programme has enabled collection of this combined pseudo-anonymised case data in a systematic way for the first time. We now have a centralised method for obtaining the number of diagnostic genetic tests performed for a specific cancer susceptibility gene and the number of times a specific variant was identified through this testing. Alongside ethnicity data from all of the NHS in England, this case data can then be compared to ethnicity matched population data from large-scale population databases like UK Biobank. Currently, this data exists for BRCA1 and BRCA2, and the Lynch syndrome genes MLH1, MSH2, MSH6 and PMS2. The ethnicity-specific Public Health England data can be accessed in full by registered members through the CanVar UK website. This infrastructure enables us to obtain more accurate evidence to try and answer the key question: is the prevalence of this variant in affected individuals statistically increased over controls? However, a key limitation in using this data is that the case control analysis needs to be ethnicity specific. This is because we know specific genetic variants, both benign and pathogenic, may be enriched with specific populations. The majority of diagnostic testing undertaken in England has been in individuals with Northern European ancestry, and case data from individuals from minority ethnic groups is not yet available from Public Health England. This highlights the importance of international efforts to gather both case and control data in large numbers from a wider range of ethnicities to offer equitable access to such evidence. So how can we use this case control data to perform a statistical calculation to provide evidence of PS4? Prevalence in affected individuals is statistically increased over controls. Let's consider a fabricated novel gene, cancer susceptibility gene 123. This table shows ethnicity matched population control data from the CSG123C.181T2C variant as if it would appear on the CanVar UK website. The CanVar UK website pulls data from the UK Biobank population database. This site also allows you to select for female-only dataset in Biobank. This distinction is important, as when considering BRCA1 and BRCA2 variants, we need to consider the female dataset, as males with pathogenic variants in these genes have much lower rates of cancer than females. As you can see from the table, this variant has not been reported in the Biobank all participants population database. For this dataset, we have the number of individuals in whom whole exome sequencing was performed, being 51,377. However, we do not have the number of alleles sequenced at this loci. As coverage at different locations in the genome varies, it is most accurate to look at the number of alleles sequenced if that data is available. If we are provided with the number of alleles sequenced, we should divide this by 2 to get the total number of tests performed. This is because each individual tested has 2 alleles at each loci in the genome. This table shows the number of cases of this variant in the UK and the total number of diagnostic genetic tests performed by individual genomic laboratories, as well as the white ethnicity data. If we go back to our key question for PS4, when considering population data in cancer susceptibility genes, is the prevalence of this variant in affected individuals statistically increased over controls? We can now use an online calculator to see if there is a statistical difference between the Public Health England case data and the Biobank control population data. The online odds ratio calculator and Fisher exact test are our chosen tools for this. The links of both of these can be found on the CanVig UK guidelines. Here you can see we have inputted on the rows property A as the number of genetic tests done looking for presence or absence of a particular variant. The columns, property B, represent the presence of disease, i.e. cases, or absence of disease, i.e. controls. It's important for this calculation that we use ethnicity matched data. Therefore, we take the 22,594 white ethnicity case probands tested, and we take away the number of times the variant was found, 7, resulting in the number 22,587 to use in our calculation. For our control group, we need the number of individuals of the equivalent ethnicity from the population data. And for this, we take the white figure of 51,377. We do not need to take anything away from this number, as there were no individuals with the variant identified within the population data. As we cannot use 0 in the odds ratio calculation, we have to apply the Haldane-Anscombe correction and add 0.5 to all values before calculating the odds ratio and confidence interval. The odds ratio outcome needs to be greater than 10 for application in high penetrance genes. The CanVig UK guidance states that for enriched family case series, such as the Public Health England data that we are using, the odds ratio for high penetrant genes or variants needs to be greater than 10. If this number is greater than 10, we need to confirm that the confidence interval for this calculation does not span 1. If the confidence interval does span 1, we cannot be confident in the statistical significance of our results, and so we cannot use PS4. However, if we have a sufficient odds ratio and the confidence interval did not span 1, we can then look at the p-value to consider if we can use this PS4 evidence category and what strength it can be used. The p-value is calculated using the Fisher exact test. As the p-value here was 0, so less than 0.0025, we can apply PS4 at a level of very strong. To consider what strength of evidence we can use, the CanVig UK group has provided guidance about the weight at which different p-values can be used, varying from PS4 supporting to very strong. For the above example, we spoke about the odds ratio required for high penetrant genes or variants. But as we know, things aren't always quite that simple. Remember this graph of how different types of genetic variation contribute in different ways to disease causation. We know that different types of cancer susceptibility genes exist, and different types of variants within those genes also exist. Some are much less penetrant, so as well as lower effect sizes, we would expect to see them more often in the population databases. To consider this, the CanVig UK guidance gives odd ratio figures to use for these less penetrant genes. It's also likely that as more and more gene-specific guidance comes out, like those available for BRCA and TP53, we will need to consult these for what values to use for different genes. These calculations, just like everything else with variant interpretation, are always updating over time, with more case control data available, as well as more detail about what odds ratio to consider. As a result, it's important to always consult up-to-date CanVig UK consensus guidance, and if you're unsure, please speak to your local experts to check in.
So, hello, my name's Claire Turnbull. I'm a professor in translational cancer genetics at the Institute of Cancer Research and also an honorary consultant in cancer genetics at the Royal Marsden Hospital. Cancer Variant Interpretation Group UK, we set up in 2017 in response to a decision by the ACGS, the Association of Clinical Genomic Scientists, that we would adopt the new ACMG framework for variant interpretation from the American College of Medical Genetics. So there was a group set up to roll out this implementation for rare disease, but it was recognized that actually how we use this guidance for cancer susceptibility genes might be a bit different, and that we needed a special group to focus on that. So that was the inception of CanVig UK, and we've been meeting monthly since 2017. The ACMG, the American College of Medical Genetics, in 2015 released this new framework around variant interpretation. And although that really set a very clear set of instructions about how to use different evidence types to interpret variants, a lot of the elements within it were relatively non-specific. So there would be, for example, evidence item PS3, a robust functional assay. What's robust? So we needed to make our decisions within our community about what constituted a robust assay. Strong evidence of an excess in cases compared to controls. Again, what did we mean by strong? Absence in population data. How large did that population data need to be? So there's actually quite a lot of work there to ensure that we took this new framework that was designed to deliver consistency, but actually the devil is always in the detail, that we ensured that we were consistent in how we were using it. I think the challenges are that actually consistency in variant interpretation is difficult, and the ACMG framework, as I said, was just guidance, but what's happened is there are lots of different channels of activity which are further ratifying that ACMG framework. So what we're trying to do is evolve this specification of the framework, but it's really within a sort of broader landscape of the rules and the goalposts continually being altered or adapted or reevaluated, and trying to be consistent and logical without contradicting these other types of evidence. So that's a huge challenge, and what we've tried to do is we've released an initial CanVig UK specification of the guidance, and we evolve that annually to try and give clarity generated from the CanVig community, but documented in a clear, logical fashion about how we would recommend application of these rules. But that is a challenge to maintain that guidance and maintain it being consistent with other groups. Another challenge, like anything, running a group requires quite a lot of organisation in organising the meetings and so forth, and we're very fortunate that we have Cancer Research UK funding now supporting the secretariat of the CanVig UK group. Well, I think it really does help us having this national multidisciplinary network around interpretation of variants in cancer susceptibility genes. We have a very lively email group, and people can just throw a question in there and they'll get lots of answers, lots of responses. It may be a simple question like, have you seen this variant in your lab? But they'll get 10 to 20 responses, which just help them calibrate whether the variant's been seen before. So we have the email group. We also have monthly meetings. That's a forum for people to bring any particular questions or issues, but we also use it. We have variant of the month. So that's an opportunity for people to have a tricky variant looked at by the group in detail, or to use it as an opportunity for education by looking at a particular gene or particular scenario or a particular type of evidence, and as a group to sort of learn together how best to apply this. We also have our third way of communicating is we have a shared MDT web tool, Canva UK. So that's where we can share our classifications. It's got a private NHS area where we can share notes, variant classifications, comments and so forth, but also a wealth of resources from different functional data, in silico data, population counts and so forth. So that's a third way that we can share as a national network, and clearly there is huge value in having this national interaction, because variant interpretation is not easy. And so the advantage of having a national network is we are getting better at being consistent. We have a means to check with each other if we think we might be being inconsistent. If there's new evidence, we have a mechanism for alerting in regard of a new variant reclassification, be it up or down. And finally, it gives us a mechanism by which to interact with international groups. So a conduit through which we can feed what's going on in the UK to the international groups. Ultimately, patients come to genetics to find out whether their disease has a genetic basis, and often this involves a genetic test. Genetic tests, you find variants and they need classification. So anything that's influencing that classification is then affecting patients. So there are a number of examples where through data sharing through CanVig UK, we have been able to take a variant of uncertain significance and up-classify it to a likely pathogenic or pathogenic. And that's clearly very impactful for the relevant families, because those who carry the variant designated as likely pathogenic or pathogenic have the management, be it screening or surgery. Those who don't have the pathogenic can be largely reassured they're not at increased risk. Conversely, there's been the odd occasion where historic data has meant that a variant has previously been ascribed as being pathogenic, but through data sharing within our network, we have been able to identify sufficient data to cause concern to down-classify that variant. So again, by having this network, we can disseminate those urgent reclassifications rapidly and ensure that across the UK, we are managing our patients with the best and most up-to-date integrated data.
So congratulations, you've got to the end of week 1. Well done. Thank you for staying with us. We've considered in this week how the genetic architecture of complex disease, like cancer, requires different considerations with respect to variant interpretation when it's compared with rare monogenic paediatric disorders. And then we've also discussed the CanVig UK national framework for the interpretation of variation in cancer susceptibility genes. And we've then just looked specifically at population data and in silico data lines of evidence, and how we can adapt those using case-based examples in cancer susceptibility genes. And we've also, as you know, woven throughout a case study with Liz and Thor, who've received an uncertain genetic result, for whom your interpretation of the variant next week will have clinical consequences. So thank you for completing week 1, and we look forward to meeting you again in week 2.